Alright, this video is going to wrap up the photosynthesis unit. So part 3 of the notes come in here, and we're going to start right away with talking about the Calvin cycle. That is the focus for part 3 of the notes. Objective 11 says to explain the light-independent reactions, aka the dark reactions, the Calvin cycle, same name, and identify reactants and products. So first of all, let's define it. Light-independent reactions, or dark reactions, also known as the Calvin cycle, takes place in the stroma, which is kind of the cytoplasm of the chloroplast. It produces glucose from ATP and NADPH and CO2, carbon dioxide, from the atmosphere. The main point about this to recognize is glucose. That's the sugar that plants are making as their food source, so they do not have to consume other things. That makes them an autotroph. And then for glucose, really quick definition for this guy. First, the chemical equation for it, C6H12O6, which is the sugar made by photosynthesis, important for all life on Earth, or just about all life on Earth. It also says we want to talk about reactants and products. Reactants are things that are obviously going in. So going into the dark reactions, going into the Calvin cycle, the reactants are mentioned here in the first definition. ATP and NADPH, those are from the light reactions. And also CO2, which you could say is just from the atmosphere. That's our waste product. We'll see in the next unit how our waste products and plant waste products are so important to each other. So going in, ATP, NADPH, in the form of these electrons. That's the important part of this molecule, is the electrons. And here's the energy molecule. Remember, ATP is the energy molecule, so that those chemical reactions can take place. It's going to spit out some products, and really the most important product, like I said, is glucose. However, going back to the light reactions, or ADP and NADP+, those are going to go back to the light reactions so that ATP and NADPH can be made again and come back and supply the energy and electrons that the dark reactions need. However, the most important part of the dark reactions and all of photosynthesis, in fact, is this guy right here. That's C6H12O6. That is really important for life on Earth, that, pretty much all life on Earth that are consuming things like plants to begin food chains. So this is where most of the energy on Earth is actually coming from, plants. Alright, so there's reactants and products for the dark reactions, aka Calvin cycle. Next, we're going to talk about the 3 parts and the cyclical nature of the Calvin cycle. So first, let's just define Calvin cycle. Pretty simple. It is a series of chemical reactions that uses CO2, ATP, and NADPH to make glucose. Series of chemical reactions that uses CO2, ATP, and NADPH to make glucose. You can pause that if you need to. Next up, carbon fixation is just the term that describes the attaching of a gas to an organic molecule. In this case, the gas is CO2. Again, our waste product. We'll see how that relates in the next unit. So, attaching a gas to an organic molecule, that's called carbon fixation. The carbon, of course, is right here in the CO2. Next up is this molecule called RUBP. We don't care what it stands for. It is a 5-carbon molecule that stays in the Calvin cycle so that the cycle continues. You can see here it talks about the cyclical nature of the Calvin cycle. There has to be molecules that stay in, otherwise it is a linear process and not cyclical. Next up is pGal. Also, you'll see it known as G3P. Remember, in organic chemistry, everything has two names. So there are the two names for this molecule. But in any case, it is a 3-carbon molecule that is used to make glucose and to regenerate the molecule that is staying in. Oops. RUBP. Alright, so let's move on to the phases of the Calvin cycle, and there are 3. They are all pretty different from each other, but they're all just a series of chemical reactions. Phase 1, again, is carbon fixation. That's where CO2 is attached to the 5-carbon RUBP. And that is seen right here in the picture. We're going to talk about the beginning of the cycle right here, where CO2 comes in on the top, attaches itself to this 5-carbon molecule, RUBP. There's 5 carbons, 1, 2, 3, 4, 5. To become a 6-carbon molecule, which instantly splits up into 2 3-carbon molecules right here. 1, 2, 3 carbon molecules, which takes us into our next phase. Creating that pGal, or G3P, that's going to come out and eventually form a glucose. This is the reduction phase, and the reduction comes into the picture right over here, where the electrons are added. You can go back to your redox chemistry. If electrons are added, it's called reduction. It's because it's reducing. Adding electrons is reducing the charge. So we add electrons to this guy that we don't really care the name of. That becomes our G3P, aka pGal. Right there, 3-carbon pGal. 1, 2, 3. Those other molecules attached to these guys right here are phosphates. And that comes from ATP. That's, what, again, the energy molecule. And then you see one of these guys is going to drop out and eventually become glucose. So eventually that 3-carbon molecule forms into a 6-carbon glucose. Just needs some more chemical reactions and a little bit more carbon molecules. Phase 3 is how this cycle continues. Notice the pGal drops out to become glucose, but there is stuff that stays in so that the cycle is a cycle. And that's the phase 3, regeneration of RUBP. So we need to create more of this RUBP so that the cycle can continue. And that's done up here. This pGal goes back in, some more chemical reactions take place, turn that 3-carbon into a 5-carbon, which then will continue the cycle once again. I'm going to show you an animation that if you go to the website, it's just called the Calvin cycle. It's pretty easy. And that's what this animation is going to give you a really good idea of how this works. Let me just zoom in just a little bit, or zoom out a little bit, so it fits on our screen. Alright, there we go. So again, this is going to take you through all the phases. Here's stage 1. Rubisco is the enzyme, attaches carbon in the form of CO2 to RUBP. So we just saw. So that's how that works. And you can see it's happening a few times. We're not going to talk about all the molecules that are made. It's not just one at a time, but not details we need to get into. So you kind of see what happens. It changes into a 6-carbon molecule that instantly splits into 2 3-carbon molecules. And they're going to collect over here when we get into the next stage of the cycle, which is converting that molecule into pGal by adding phosphates, and then removing the phosphates using NADPH and electrons. So it's just a simple matter of making stuff and breaking it down, and eventually we spit out these G3Ps, better known as P-gal, and possibly easy to remember. One of them is going to come out, and that's where the glucose is going to get made. The rest of these guys stay in, and that's our last phase, regeneration of RUBP. And again, it takes energy. There's the ATP going in to the system and spitting out those RUBPs again. So from the light reactions, here's our ATP and NADPH that were used by the dark reactions eventually to spit out a glucose, and most importantly spit out a glucose, but also to keep this cycle going so that more glucose can be made. Back to our notes. Reactants and products of light-dependent or light reactions, and dark reactions, light-independent. Sorry for the headache. So here's our reactants and products, which you've already seen before. Go ahead and pause the video and write those down. This also brings up a good time to talk about the leaf disc assay lab that we are doing, in which photosynthesis takes place, causing those discs to rise. But guys, things to focus on are right here. Things that are going into the dark reactions, coming from the light reactions, that's ATP and NADPH, right here. And then they're going to be used up and spit back out in the form of ADP, NADP+, going back to the light reactions after the dark reactions is through with them. And one of the most important parts, right here, the whole point of photosynthesis is to make glucose. So plants have their own food source. I'm going to leave that picture up there with the floating discs to bring up some good points here. Factors that affect photosynthesis. Well, first of all, let's think about what photosynthesis is using to actually do its job. That is sunlight, CO2, and water. There are other things that can affect photosynthesis, obviously, like the time of the year for trees that lose their leaves. That's going to affect the rate of photosynthesis. Even time of the day, the temperature, amount of wind. There's a lot of things that go into it, but just some basic factors that we can talk about just straight from the equation are right here. And especially these guys that you can control in a science experiment, even sunlight, but really you'd be just using artificial light. So more of these things is going to increase the rate of photosynthesis. Less of them will decrease the rate of photosynthesis, which, again, you'll see in the leaf disc assay lab. The bicarbonate was the carbon source, so as CO2 is actually bicarbonate, as baking soda and water. They're sitting in water and then applying a light source. Not exactly sunlight, but artificial light is just as good. So that finishes up the photosynthesis notes, focusing mostly, again, really good thing to do is to go back and watch this animation right here of the Calvin cycle so that you can understand the process completely.